slide because it's a good highlight of some of what we're interested in. So you can see the roots of this young corn plant from a 3D gel imaging system or optical projection tomography. And this green stuff here is through positron emission tomography. So this is a functional imaging method. This is work at the med school. We can feed a plant a radioactive carbon dioxide and literally watch it as it photosynthesizes it and decides, so to speak, where to move it around, allocates the carbon both above and below ground. And so these are the types of tools that we're trying to approach some big questions with. So tell you more about that. I won't go over too much of the motivations because I think this audience understands it well, but going back to C.M. Donald's 68 paper on plant ADT types and thinking about characters of green revolution wheat, there's sort of a big hole here. And I think roots were mentioned once or twice in Donald's paper, and we really still have not filled in this, this hole here. And in particular, I'm interested, my background is in genetics, and in particular very interested in genetic control of root growth and root architecture. But we need good tools to do that. If you measure something complex with a simple tool, often you don't get to the answer. And so to some extent, we're engaged in all of the many myriad functions and that roots provide, and the potential means to meet some of the major challenges around agriculture. So I think many are familiar with this term, but root system architecture, simply the spatial organization. I think you could add temporal and spatial organization of roots in the soil. Classic image from Heidi Nanchura, but basically after John Weaver's work, the different prairie grasses that grow in my part of the world, Missouri, and further west into the prairies. And you can see the diversity in form. And as we know, the architecture of the root system has profound implications on the ability of that plant to capture water and nutrients and compete for these things in ecological and agroecological settings. Of course, here's the problem. This is how these images, this is Weaver and his team 100 years ago, digging these roots out basically on an archaeology expedition. And, you know, this isn't literally my team, but I think we're all fairly familiar with this. 100 years later, we're still doing the same things. We really don't have a magic machine that can go over the field and see the roots. And so there are significant limitations in the ways that we can look at roots, and these affect what we can actually know about them, how we interpret the biology, certainly how we get to the genetics. And so a lot of what I'll be telling you today is our approaches to getting more information using the tools that we have, and continuing to work and developing the tools, the sort of next generation tools, which are won't be easily come by. You know, here's why it matters. Just one example, and in one of the few cases we've actually identified the gene controlling a quantitative trait in a crop plant. This is in rice. IR64 is a widely grown breeding line that goes back to Green Revolution rice, but it's known to be fairly drought susceptible, and it has a fairly shallow root system. One base pair mutation.
Is this paddy rice?
This is
Upland rice.
This is upland rice.
Okay.
Yeah, this is upland rice.
Not grown in water.
What's that?
Not grown in water.
Yeah, not grown in water. That's right. Right. Great question. Right. You don't need the same kind of root system if you're soaking in a nutrient bath. And so there's been problems with this, and this is a staple of tens of millions of people. And so it's been a improved drought tolerance has been a target of rice breeders at Erie for quite a long time. A one base pair mutation that came from a landrace variety in this gene, deeper rooting 1, fundamentally transforms the root system. You can see the architectural changes and just the biomass change. Turns out there was a stop codon in the breeding line here. So if you ameliorate that mutation, you get a huge benefit, basically a huge yield benefit under dry conditions. And in many conditions, there's not a yield penalty for this. So this is just an example of what I think is the tip of the iceberg. And if we can really understand root genetics, there's a lot more success stories like this. So again, looking at the root architecture, this is a two-dimensional image. Obviously, this is also 3D. Hundreds of thousands of roots in mature maize plant, several hundred cubic feet of soil that they explore. It's been estimated miles and miles of roots on a single plant. It's phenomenally complex at this level. But then, of course, really this root system is a property, maybe an emergent property, or maybe something we can actually really understand. But each root tip is exploring a microenvironment and engaging with microbes and chemical interactions and physical interactions. And so how the processes at this scale manifest at the larger scale is a really big question that I think we're going to need to come to some satisfactory understanding of in order to achieve a lot of the goals that we wish to, in terms of leveraging roots to improve agriculture. So one of the sort of obvious places we could start is root gravitropism, and I'll tell you a little bit more about the genetic work we're doing, but we've been working with some agravitropic mutants. This one is ADT Star in corn, and you can see the roots basically grow. Whichever way they start growing, they just keep growing. Now, this is Adam Braze, who's a grad student at University of Mizzou, but he's in my lab through an adjunct setting. And so each root tip, of course, is sensing gravity, and it's doing its own thing. It's being agravitropic, but this eventually manifests itself as a root system architecture. So we can start to connect the dots a little bit here, right? It fundamentally changes the steepness, so to speak, of the whole root system. So that maybe gives us some entree into understanding the relationships of the two. Some of the tools we have to look at this, and again, to really quantify the root system in a way that we haven't been able to before. This is the gel-based imaging system. And so I saw Dave's today. It's fantastic. We have a very similar system at Danforth Center. It's just growing the plants in a gel and spinning them around and capturing the three-dimensional architecture. We then turn this into an actual three-dimensional model of the root system. And this is, again, very powerful because we have the explicit 3D architecture now modeled. Unlike in a field where you dig with a shovel or you take a soil core where you're taking some probably sparse estimate of the actual root system, here we have the root system. And now we can measure a lot of things about it that can't easily be measured in other ways, including the topology of the root system. And there's a whole bunch of ways that we approach extracting information from these shapes. And I'll touch on it a little bit, but essentially, when we have a better model of the root system, we can measure much higher information content traits or phenotypes from it. Additionally, in this system, we can measure dynamics. So when we talk about interactions, that implies, of course, growth and implies dynamics, but often we can't really measure these very well, or we pick arbitrary time points because it's experimentally possible or experimentally impossible to capture the true dynamics. Here we've automated the system, and you're looking at root growth 24/7 for a week, every 4 hours, even during the night. And we were able to, these are two maize genotypes, are pretty well-known genotypes. And from this, we're actually able to take the 3D models at each time point, develop the software to overlay them, and we can now measure each individual root in this complex growing root system, right? So these things start out pretty similar. They both have a primary root. But that architecture changes quite a bit, and that's being driven by the genetics. It's being driven by the sort of root carbon allocation and developmental mechanisms and the differences between these two maize inbred lines. And even after a week, the architectures get pretty distinct. In the middle here is the hybrid between them. They get pretty distinct quickly, and so what we're able to understand here is how decisions at the local root growth level manifest again as different architectures. And we could do this pretty explicitly here. So I won't get into all the details of this. It's published now. So, and we were able to track a lot of the features of individual root growth over time and draw that back into the overall architecture. Similarly, we were able to use a simple logistic function model to understand the growth not as a series of measurements at different time points, but as a function. And we can use the values of that function as the phenotypes themselves. And so we have basically three functions. They're alpha 1, 2, and 3. Alpha 1 here essentially describes the carrying capacity. So this is the maximum, in this case, total root length of the system. And it turns out they're in the end, they're not statistically different. They have a very similar total root length, but it's apportioned differently, and it gets there in a different way. You can see that here with alpha 2. If you take the first derivative here of B73 and Missouri 17, both of them increase their growth rates really quickly, and then they both hit a peak. And then they're still growing, but just slower than they were before. Missouri 17 is a little more flat, and that peak is offset here. And we can capture that with this parameter alpha 2. And alpha 3 gives us basically the slope, so how quickly it reaches the carrying capacity, which again, you can see here to some extent in the slope of that curve. So it's really giving, again, a function value that's supposed to be a little bit more comprehensive in terms of the root growth dynamics, rather than picking arbitrary time points to measure. An additional part of this, and again, the caveat here is this is what we found in this work. I'm not saying it's universally true, but it was a really nice finding, I think. I'll tell you a little bit more about how we get the 3D root crown architectures from the field, but basically we dig these up with a shovel, so it's about that size. We wash them off, and because they're lignified, they're stiff, they maintain their shape. We scan them with an X-ray system. And so now what we're able to do is actually measure 3D traits in a mature plant from the field, and measure the exact same traits with the same information content from our gel system. So we're not comparing apples to oranges, right? We're using the exact same types of measurements on both. And what we found was that basically the genetics rule. So the genetic signal can be seen both as a function of development, because these are much older, and also moving from this artificial system to a field at Mizzou, if that makes sense. So the field of maturity or gel grown clustered together here, and same thing, they're very well discriminated along this principal component. And if you look at any one of the single traits, you get the same pattern. If one genotype was bigger as a seedling, bigger in the field, vice versa. But you can kind of see it all here. The hybrid's a little funny because here we don't get hybrid bigger, but here we do. So these in the field here, these are just much bigger bigger plants. That makes sense. So again, our approach here is to really understand genetics, and we look at just genetic variation. So these are maize varieties from mostly around the US, but also around the world. These are from Simit. This one's from Thailand. This is a Hopi variety, a landrace. And so these are part of the nested association mapping founder lines. And you can see by eye that there's differences, but the trick is actually turning this into quantitative information that we can use to understand the biology. So this is our field operation, and it started again with me in a pickup driving up to UIUC, about 3 hours away. We do a lot of our field work, and asking Steve Moose if I could dig up his old roots once he pulled his ears off of the Illinois long-term selection. He said yes, and he got his whole lab out to help me. And so we dug up a bunch. And so this turned into now, we do many thousands of these every year. We have got a trailer, we drive around wherever, and we dig up the roots. It's humane, it has air conditioning and everything in it. You can see here, Shayla is, she's got a little imaging system, and so she's taking these formal portraits, so to speak, of the root crowns, and using a software that Alex Book, she's a professor at my alma mater, my old department, University of Georgia, developed, and it's on Cybers now. So basically, read these papers. You just need to take sort of a portrait that looks like this, and you could upload them to Cybers, and you can get a lot of information about the root system through computer vision image analysis, right? And it's entirely free. It's a really nice system. There are other softwares that do similar things. So basically, we no longer have an excuse not to study the root system, right? At least to some extent. It's quite easy to do now. It's quite straightforward. So one of the projects we've applied this to is trying to understand G by E response to low nitrogen. So this is a paper we've been working on forever. It's still in prep, so it's sitting on my desk somewhere. But this was also a project with Steve Moose and his high and low nitrogen fields. We took a series of inbreds, crossed to 2 different testers, grew them in high and low nitrogen, and measured sort of whole plant responses or not across three different years. So you can see a few nice trends that are very consistent. For example, the root biomass
increases in low end. That's very consistent every year. In high end here, you can see there's some variation from year to year, but there's a very clear trend across all this germplasm. So that was nice to see. We expected this, but it's experimentally validated. I'm not going to go through the whole story here, but I'm going to give you sort of the punchline of this paper, which is roots are quite plastic, and they're actually quite hard to explain, even when you have like fancy image analysis methods, right? So if you look at the, we also combine this with ionomics, so seed ionomics with Ivan Baxter. So this is the variance explained. This is a residual term. This is due to N levels from an ANOVA. This is due to genotype. This is normally turned the other way, so I'm having a hard time interpreting. But as you can see, you can get quite a bit of variance explained by the nitrogen treatment for the above ground traits, which are these blue ones here. A good bit of it by genotype. There's a little bit that's unexplained, but that changes dramatically with the roots. And for most of our variation was just not explained very well. And it could be the experiment. It's not that it's impossible to do this kind of work, but it was a fairly robust experiment. And so there's a bit of a word of caution not to overinterpret 1 year or potentially even three years of root phenotype data. There's general trends that I think you can discuss, but any one of these particular root traits, a lot of it was quite hard to interpret. So I normally don't talk about this bit, but I did just want to sort of get that out there this time. Here's a little bit more solid ground and work we're doing. And again, the idea here is to leverage the power of genetics. So the Illinois long-term selection experiment is one of the longest running genetic selection experiments, certainly in plants, if not all of biology. And so there's two parts to it, an oil and a protein. And we've been working with the oil lines. Again, this is with Steve Moose. And so basically back in the late 1896 or 8, there was a recurrent selection scheme for either high seed protein or low seed protein. This is an open pollinated variety, Burr's White. Founding population is apparently a few hundred individuals, right? This is before hybrids. This is a long time ago. And every year they took the top and bottom seed protein cobs or kernels and planted them out again. And they've driven seed protein down now 100 plus years later, almost. This is the minimum you need to germinate. High protein has continued to go up, which is a phenomenal genetics or epigenetics, I don't know. But the reason I got interested was because these plants are very good at capturing nitrogen. And so you would expect, even though root traits weren't selected for, that there had been indirect selection for this. And so we could see that really clearly early on in the gel experiment, big difference between inbred lines that were derived from these two populations. We can definitely see differences in the field. And so there's a intermated mapping population between these two. And this is where we got started doing like really big, large scale root phenotyping. This is in collaboration with Ivan Baxter with ionomics, but Andrew Leakey, this is his field at UIUC and his drone. And these white things you see here are mini rhizotrons. And they're like thousands of these things that he's pounding into the ground. These are about a meter long, pounded into the ground. In this case, we have more. In this case, we had 3 to apply. Now I think we tend to do 6 to get better statistics there. But the idea was to understand information that we can get from a mini rhizotron, which is something. It gets roots at depth, but it's not a great estimation probably of the real root system. And then combine that with our root crowns. So we'd like to understand the correlation at the data level, but really at the genetic level. Can we identify genes or loci in common through a GWAS hit that from both root crown analysis and from the soil access tube analysis. And so we actually have been able to get quite a lot of good information from this. If you look at, so this is the soil and this is depth. If you look at IHP here, which is good at capturing nitrogen, and ILP, you can see there's much more biomass and it's at a much deeper depth, or a significantly deeper depth than ILP. And these are just extreme. So we get a lot of variation within this mapping population, which is what you want. That's how you can actually identify QTL. So what I'm going to show you here wasn't found with the mini rhizotrons. It was found before that. You get an idea of at least how we're trying to combine sort of the best parts and mitigate the negative parts of the available root phenotyping methods that we have. by joining these things up. This was found just from dirt imaging in 2015. This is the result of a GWAS, and we found several genes, but in particular, this candidate SNP here. This is Zhengbing Liu, who's a postdoc in the lab. He's named this thing ideal root architecture. Very optimistic. I don't know if it's ideal, but it does something about roots, right? And so basically this gene here, we've identified, and we've now found that there's a mutation, the promoter of ILP, and so it changes the gene expression. And if you're an ILP, if you have the ILP allele versus the IHP allele, you get these expression differences. So we think this is this mutation, changing gene expression, and it's providing a difference in the root architecture phenotype. So we think we've identified a gene that controls quantitative root architecture in maize. I'm not going to tell you that much about the gene itself today, but is in the central carbon nitrogen metabolism pathway. So we think what's happening is in ILP it's broken, and so the gene product or the metabolic product is being shunted towards something that the plant interprets as nitrogen. So the ILP, even though it's N-starved, thinks it is N-satiated, and it grows its roots accordingly. So this is not something directly affecting the development of the roots, but it's something affecting the plant's decision-making process, if you will, to grow roots in a certain way, or foraging algorithms, or however you want to think about it. And because this gene is quite conserved, we see a very similar thing in Arabidopsis. There's 2 orthologs, and one of these, IRA1, has a very strong root phenotype. So we're following this up with some metabolic analysis, which is getting me back to like biochemistry 101, because I forgot all the CN cycling pathways, to be honest with you. So anyways, you never know where your science is going to lead you. It might lead you back to the thing you snoozed through as an undergrad. So pay attention. So this is a little bit more, this story is a little bit more comfortable for me, up my alley. This is back to maize genetics. This is a project of Adam Bray. This was a mutant found in 1930, published by Jenkins, found in a farmer's field. Jenkins published it. And you can see here, it makes almost no nodal roots. So these are the roots that form at the base of each leaf, and they become the predominant root system of corn. And they form in a whorl at the base of each leaf. And in rootless 1, the most severe mutant, there's almost none. And there's definitely none above ground. Very strong mutant. We know now that it's a VHLH transcription factor, which is pretty cool. And it's a classic maize genetics story. So we were able to verify this first with a couple of transposon insertions. So we have a DS transposon insertion here. There's a wild type, and you can see that has many fewer nodal roots. So we're pretty sure this is the gene. We have a second allele here. And then it was very hard to clone this because there was a classic maize genetics, a bunch of transposons stuck in here. And so we actually had to use this nanopore sequencing, which was super cool. I don't know if you guys have done this, but it's a little USB drive, and you make some high molecular weight genomic DNA, and like overnight, we basically lightly resequenced the whole maize genome, and we got one read that went across here and found these transposons. It was really cool. So anyways, we were able to identify this gene, and it's definitely this BHLH transcription factor. And here's the original mutant here, RT1, that's got these transposons. And it turns out even in the wild type segregant, because again, this is 1930, so this is old germplasm at this point. This thing has its own little transposon in it, and actually has more than the normal number of nodal roots. So that got us thinking that maybe this is more than just a loss of function, gain of function mutant, but actually something that we might be able to use to tune nodal root architecture, right? Again, and that would have a profound implications for our ability to sort of develop root systems that were suited to various environments or various agronomic purposes. So again, very similar story here where the gene expression is quite low in RT1. A really cool finding recently, we're like, well, what is the actual like causal, like developmental problem with these nodal roots? And so working with Josh Strabel in the Scanlon lab, who's an in situ master, we've now found this really awesome pattern. This is a crown or nodal root primordium in an early seedling, and we can see this really great pattern here. This is very reminiscent of scarecrow or short root, so we think it might be involved in endodermal cortical patterning. A little bit earlier on, we actually see this in the incipient primordia as well. So it's in a fundamental like role in nodal root development. And these are just some controls for good measure. So the interesting thing though is if you look at nodal roots again across like a panel like this, there's quite a bit of quantitative variation for it. And then we're reading the literature. I'm telling this not in the real order it happened. We already had an idea about the literature here, but RT1, once we knew where it was on chromosome 3, turns out there's actually 2 QTL studies, one with a teosinte and one with this weird Gaspé flint line that Roberto Tuberosa and Silvio Salvi have worked with, where they identified a quantitative trait locus that was, really overlapped RT1 pretty well. And so we're starting to suspect that maybe this actually could control quantitative root variation. And in fact, in a couple of integration lines of teosintes, this is one teosinte integration line, this is another one, we actually see like this weird patterning. So normally the nodal roots are very consistent. They kind of line up like this. You can see these like little ones that got kind of stuck in between those. And these actually make more nodal roots than normal. So we don't really totally understand this yet, but we do think that it is controlling quantitative trait variation. So we've done a couple years of field experiments and found that if you have the teosinte allele, the problem is these are messy because they have other teosinte stuff. These are various integration families, but on average, if you have a teosinte allele versus the maize allele, you have more total crown roots there. Yeah, here's another example of that, just with a different teosinte population. Actually, this is an integration line, so this is only the region of RT1. Everything else is maize, and it boosts the crown roots there. So lots more to do here still, but I think we might have gotten a pretty good piece of the puzzle there. Okay, so I'm going to transition a little because I promised you spinning roots, and I don't want to get too late without getting into these spinning roots. Two of the technologies we've been heavily invested in, or a general technology, is Xray Imaging, and we have 2 platforms. This is an X-ray tomography system, an X5000. This is basically made for aerospace industry, so it's really nice. Like 2 or 3 of us could stand inside of this and have a conversation. Unless you're like really tall, you basically it's quite comfortable in there. So it's made to put airplane parts in. So we can put like whole plants in there and scan and see inside of the plant. And then we have an additional platform, which is an X-ray microscope that, as I'll show you, lets us get down to cellular resolution. So again, the idea of being able to follow the developmental trajectory of an organ, of a single root at the cell level to the large architecture is what we're after with these. And Keith Duncan runs these things. He has a great Twitter account at CygnusX1, which is the first, there's a black hole that was discovered that makes a lot of X-rays. So that's what he named this thing. That's Perseus. Okay, which is another astronomical thing. Okay, so these X-rays here, hopefully this gives you a size of scale. It's all automated, so we can basically move so the way X-rays work is this thing shoots a beam of electrons at a little tungsten target right about there. There's a very small aperture. It's a 6-micron target. It makes a cone beam of X-rays. These X-rays pass through whatever it is that's in front of them, and they hit the back of this detector and they form an image, which is called a 2D projection or a radiograph, right? So when you get an X-ray at the dentist, it's just doing one of these. But the tomography part is where we spin the plant in this case, right? The medical imaging systems is that big donut and the things spin around you because you don't want to spin the patient. In this case, the guys at North Star call this a part. They will not call it a plant, they will call it a part. They call it a patient at the med school. It's funny, but anyways, this allows us to then, again, get a 3-dimensional reconstruction, very similar as the optical system, but this is growing in a much more realistic substrate. So we've done this work in soil, but typically we work in turfus or another clay substrate just because the methods development is a lot easier to do that. It's certainly possible in soil, it's just harder, like most things. So I just want to point that out. It also allows us to look at organs, like for example, this developing ear. So I can count the number of husks that will be on this ear quite early before you can see any morphological change outside macroscopically. With the X-ray microscope, we can then take a sample, like this is what we dig out of the field. This is a shovelome. This is a sorghum root. And then zoom in to like a root tip or this nodal plexus, which is, again, this is where these nodal roots are coming. You can see the primordia there, and basically get information at the cellular level. You can see all the vasculature. You can see individual cells here. These little spots are nuclei. So that really gives us a wide range to understand how the complexity builds across different scales. So this is what the inside of the X-ray microscope looks like. The reason it's called that is because it uses X-ray beam here the same way. It makes a cone. And we can use a standard detector, but you can also see these microscope objectives. So there's basically a scintillator at the surface of this where your eye would normally go, and it collects the X-rays, it turns them into photons, and then it uses normal microscope optics to enhance the resolution. So we get both geometric and microscopic resolution enhancement. So again, we've done a lot of work with root tips and also inflorescences. So these are Soteria, which is a grass, model of grass inflorescences. This is a soybean flower. You can see the pollen grains and meiotic tetrads. This is a maize anther. This is what I studied as a grad student, mitosis, meiosis, centromeres. So I spent many hours squeezing these things out into, this is unsafe actually, in a little dish of paraformaldehyde under the microscope, squeezing these things out, put it in a microscope. So it was really delightful to be able to sort of see them in situ here. So this is mostly just show and tell, just to show you some of the capability we're developing. But these are no longer living. You can do living things in this system, but to get cell resolution, these are fixed and staying. And Keith has a long history in EM microscopy. So we think there's a lot of opportunity to actually start to really go back and do look at developmental biology in a new way. So again, this is a soybean flower. You can count the pollen grains. We're seeing seed development in sort of a way that it's very difficult to see. So EM, SEM resolution, but it's a 3D model and it's intact. So really just, you can tell I stare at these a lot, but I mean, it's pretty phenomenal. So these are movies, but of course we then have to turn that into data. So this is what we call segmentation. So training a computer to identify object of interest from background so we can measure that specific object of interest. So this is Ni Jiang, who's a really talented, she's a senior computer scientist in the lab. And she started to work with some of these materials so we can start to segment out different parts of these images, and then we can measure various attributes of this over time. So here's another one. I mean, we could sit up here all together and count all the cells in this thing. Keith made these animations, so I'm not exactly sure what we'll see, but anyways, you get the idea. It's really appealing to be able to go from, look at cellular organization and dynamics here, and then see this as it manifests into the macro structure. Another example here are these Soteria panicles. So we can see the branching structures which go on to form the floral parts. And here the colors are meaningless other than if it's one color, it is one sort of segmented unit. So we'd like to be able to measure each individual branch on this over time. In particular, we'd like to know, we can isolate these, and we'd like to know which of these is going on to become the bristle meristem, so that pointy thing at the end of a Soteria flower, and which one will go on to be the floral meristem. So we don't really know what we're measuring here, but the idea here is that we would see the incipient meristems in the floral meristem. This doesn't have as many, it's just a long branch. We computed local curvature, and you can kind of see where the meristems are forming. It is very preliminary, but that's the sort of the direction we're taking here. Same thing with maize root tips. We can look at the tissue level and measure each of these individual tissues, and then also, so I have to press the button here, but you get the idea. Actually segment out each individual cell, and there's some noise in here. This isn't perfect by any means, but being able to actually work with the data and segment it like this and measure each piece is critical. Beyond just the sort of animation itself. Okay, so something that is currently a lot more practical to the program is, again, taking these root crowns and extracting more data. So I showed you this 2D method, which works really well. There's a lot of information that you lose when you take a 2D projection, especially of a complicated object, right? If it's a leaf or something, or a seed, it's probably fine. But if it's a really complex structure like a root system, or especially a mature corn root system, much more difficult. So we've gotten pretty good. In about 3 minutes per scan, we've scanned many thousands of these now from the field. And again, they hold their shape pretty well because of lignin. And we've basically, same idea. We've developed a suite of algorithms that measure, again, this is a computer science interpretation. He turned all of these upside down. So just turn them over in your mind, you get the idea. But we can measure things like biomass distribution, convex hull, and the distribution of biomass as a function of depth. And this gives us a lot of ability to discriminate these complex shapes. So in fact, we can measure there's a whole laundry list of stuff that we can measure here. In this project, we're tying this back to a very simple field-based measure called root pulling force. This is very old school. You take a dynamometer to the stem of a plant and measure the force that it takes to rip it out of the ground. This is in collaboration with John McKay, and Ray Shao is a postdoc in the lab who's done this work. But basically, we were able to correlate with pretty high correlation values of root pulling force to a number of 3D traits. And so that gives us some idea of what we're actually measuring with this root pulling force. But in general, we want to use this to discriminate genotypes or genotype by environment interactions. And so we take this, again, this laundry list of phenotypes, which each one of these to me isn't particularly interesting. What's interesting is using them in a multivariate statistical way to get better and better discrimination of these complex shapes. So these are different maize inbred genotypes, and there are 4 samples of each genotype. And these are linear discriminant analysis that used all the information on the previous slide. So all the stuff we can measure out of a complicated 3D root shape, and basically what is the axis of variation, multivariate axis of variation. We can only really think about 3 dimensions, maybe 4, if you're a mathematician who's practiced, looking at tesseracts or something. But once we get to 5, we really can't do it. And if it's 30 or 100, we really can't do it. But statistics can. And so this linear discriminant is basically finding the axes of variation that best discriminate on these categories, which are genotypes. And you can see we can do it quite well here with this information. A 2D image, it's not as good because the information isn't as good. With a ruler, it's really not as good, right? Another approach we're taking, which I'm not going to get into because it's an applied mathematics method, but if you ask an applied mathematician like Mal-Yi to look at a shape like a grape rachis or a root, she doesn't parse it into branches and angles and things like that. She takes a very different approach. She takes a mathematical function, in this case it's geodesic distance from any pixel to the base, and applies this and filters this mathematical function over the shape and comes up with something called a persistence barcode, which is a representative barcode, a barcode that represents the shape. And you get a barcode for each of these samples, each of the rachises, each of the roots. And she developed a statistical way. It's called the bottleneck distance. She didn't develop it, she applied it for this purpose, where we can take any 2 barcodes and compare them to one another. It's very similar to like principal coordinates analysis, right? You take every point and you get the distance, and you want to minimize the distance between them. And so the sum of the distances is basically how different those shapes are. So we can compare now different roots or root and shoot topological shapes to one another. And we've used this for a few different purposes. So again, this one's not out yet, but you can look to a recent paper we did with grapevine inflorescences. But the idea here is all of the things that we can think to measure because we think they're biologically, or we know they're biologically important about roots, doesn't tell us the whole story. And in fact, there's a lot of information about the shape variation in the phenotypes that we can capture through other means. So increasingly now there's a method called latent shape phenotyping, which is a machine learning based method. Persistent homology is in that same category where you don't define the feature ahead of time. You let the statistics tell you what the salient features are. And so, for example, we found in this maize root study that if we take our persistent homology traits and we take that list of things I showed you with the spinning 3D root, we find 42 QTLs with these univariate traits, but when we take persistent homology, we find an additional 60 that we didn't find with the univariate. And if we take the univariate and persistent homology, and we take all these univariate and do a multivariate analysis, again, we get some overlap, but not much. I actually expected we would get more, but we didn't. So the idea here is that we're expanding the phenotypic sphere, so to speak, right? Phenotype is a lot more complex often than the tools that we have to measure it with. And so we're trying to expand that sphere so that we can understand the genetics better. That's what this is all coming back to. So I apologize, I'm not gonna be able to talk too much more about this. But anyways, if you have questions, I'm happy to answer, and we do have a little bit of this workout. So with the great paper now, this X-rays are really, really good for this. So we've been starting to do some work on great berry and cluster architecture. So again, with the X-rays, we can scan these pretty quickly. We can measure the berries themselves and measure differences in the berries. And we can computationally remove the berries and measure the rachis structure and understand how the fruit is constrained or aided or whatever by the backbone of it, by the structure itself, and how, again, this is a function of different grape varieties or different management practices, et cetera. And then eventually, the idea is to model these. So if we get good enough information from the previous work, we ought to be able to make predictive models that allow us to actually, like understand phenotypes that are to be. So we've done similar work with grain sorghum. So again, amazing diversity for this, right? But if I handed this room this basket and said measure the differences, it's hard to do. We don't. Our tools aren't that good for it. With our CT system, in 10-minute scan, and we've now done thousands of these, we can get a really, really high information content look at this complex 3D shape. It's the same idea. So we've looked across, this paper is in review now, but we've looked at the different major botanical races of sorghum and did what I would call a computational dissection. So we can identify each seed, we can measure the seeds themselves, the 3D shape of the seeds, we can measure where they are in 3D space, right? In terms of the entire panicle, measure the branches themselves, and basically break this thing down into all the constituent components in an automated way. And interestingly, what we learned about this is that while race itself has a strong genetic base, so this is the kinship matrix based on SNPs. So you can see each race has a nice little square, meaning these are genetically identical. But when we look at the phenotypes, the 3D morphologies, we don't really see those patterns. In fact, there's a significant but not very large correlation between these two. So what this means is if I handed you any given panicle and you might think, oh, that's a caudatum or that's a dura, your odds of guessing it like that are probably pretty low. With our 3D phenotyping, it turns out that seed shape itself actually is the best predictor rather than the whole panicle morphology. So that improves our genetic relationship a little bit, but it's still not all that impressive. So anyways, that's the type of thing that we're able to approach with methods like this, while we're going through all the trouble. We've done quite a bit of work also with soybean, again, looking at this multiscaler thing. So this soybean plant is growing in a turfous substrate, or actually expanded clay. And the trick is really, with the X-rays, you see everything. You don't just see the roots, you see the roots and the soil. And Dave's nodding his head, and others who've done Xray imaging. You get more than, you get what you ask for, which is like everything. And so it's quite a challenge to again, train a computer to learn what is root and what is soil so that we can measure just the thing that we want. And if you look at across different substrates like soil and turfous, and this is, I think, probably sand or something, potting mix, this is a 2D slice. It's very hard to pick out the roots even by eye. So we've been taking machine learning based approaches. This is a deep learning convolutional neural network method called UNet. It doesn't matter how it works, but we manually train data sets where we've identified the roots, and it's hard work and it's painstaking. But if we get a good training set and feed it to a machine learning model like this, our ground truth actually, it's hard. So manually, we can't even get all the roots because it's too tedious and they're quite small. And we've developed the model over time, and now in a few hours, we can extract really probably the entirety of the root structure. And then in soybean here, we can then now easily identify where the nodules are. I think the slide's not showing up that well, but we can see the nodules that are forming over time. And then the idea is to zoom in at yet another scale and look at the cellular structure of these to try to understand how the patterns we see here are related to the processes that are happening at a microscopic scale. So again, this is just segmentation results of that. And we haven't done a lot of this, but the idea here is, of course, to tie this to something functional. So this is a mass spec-based imaging method called SIMS or nanosims. And here the mass spec was tuned for nitrogen. So we can see here the subcellular nitrogen patterns within the bacterial cells here, and ostensibly the transfer into the plant cells, that being the idea. One other example of this root microbe interactions, of course, are AMF hyphae, which are quite beautiful structures that we know most of what we know about them through confocal 2D imaging or EM-based imaging. With the microscope, we're actually able to start to see these patterns in 3D. And I don't know how well you can see this, but here are some spores, and they're the very thin threads, which are the hyphae. And so these spores are actually inside the cell itself. So I'm just interested, obviously, in kind of shape and structure and the sort of infection pattern of these arbuscules. And I think I'll skip over this. This one's maybe a little easier to interpret. So here's just a 2D slice, and you can see the cells of maize root and all these white structures, which are 2D slices of the arbuscules. But of course, we have the 3D object itself. And so we can actually capture this three-dimensional structure of what is a plant root and what is the arbuscule itself. So this is not the world's most beautiful segmentation, but this is the idea. These are all of the fungal structures within the sample we were looking at, right? And of course, conversely, we can capture the plant itself through similar means. So I'm probably close to running out of time. There's a few more things I want to show you guys. Sorry about this. If you need to leave, please do, but I'll try to finish these because I'm very excited. So again, getting back to this PET, the idea here is we not only want to know the structure, but we also want to know what's happening, right? What's driving it, or what's the implications of these differences in structure we see. So again, getting back to this PET, we're feeding it a radioactive isotope, C11. It has a 20-minute half-life, so it decays very quickly. And when it decays, it makes these two energetic particles that can be detected by this ring that I'm going to show you in a minute. And that's how PET works, basically. There's lots of other isotopes you can use, but C11 is really handy because you just feed it to the plant and it does what it does through photosynthesis, pumps it down into the root. So it's a pulse chase. We give it 5 minutes usually, see where the carbon goes. In this case, there's a few pulses. That's an imaging artifact. And you can see in like 30 minutes or so, the plant has taken the CO2 up, fixed it into sugar, and it's already down in the roots, just sucking carbon out of the air and pumping it down. Here's what the inside of my collaborator, Yanshan Tai's PET system looks like. So it's a ring of detectors, coincidence detectors. He built it inside of a confinement growth chamber, nuclear physicist, but a visionary. So he appreciates plants. He built one of these in a growth chamber. And so we've been doing a lot of really neat work with it. So again, here we can use this over time. You get about 2.5 hours of carbon allocation for 3 different subjects. That's what they call them, not patients, but subjects at the med school. So we can see the carbon allocation dynamics over time, but because it's a short half-life, we can do this every day too with the same subject. And so we can actually see across developmental time how the carbon allocation patterns are changing. So you can see very early on, there's a root system, but none of that carbon is going to the root. And actually, here's a better example of this. You can see it. There's a root system, but none of the carbon is going early on, except for this one little spot, which is this shoot-derived nodal root here. That one is getting carbon. This is on day 6 after germination. 1 day later, I suspect after the maternal carbon stores have run out, we start to see the carbon moving down into the root. So just give you an example of some of the insights that we can get about these sort of underlying processes that are driving root growth. So now that we have the X-ray system, we're doing more of PET CT. So this is not the most beautiful, but it's a soybean root. And you can see these this here and that here. These are nodules. So we would like to be able to measure explicitly how much carbon is going to each nodule. And with a nitrogen tracer, we can measure the reverse using N2 gas, how much nitrogen is going back into the plant to really measure these dynamics. And it's quite quantitative. So the final example here is Medicago root system. So we would take a Medicago root system, scan it by X-ray. You can't see most of the roots, it's just washed out. Follow the carbon over time, and then we'll start to see this hotspot. So these have been infected with AMF fungi. And the idea here is we identify this hotspot and then cut that piece out and look at the microstructure with the XRM and possibly with SIMS. That being the big idea. So in the very last section, I'm going to zoom way back out. This is our version of the mesocosm systems, and Dave's are amazing. Ours are much less sophisticated, but they're bigger. And so the idea being here is that we outfit a very large box with a lot of sensors, and we can measure the dynamics over time. And then at the end of the experiment, remove the soil and capture the full three-dimensional root structure. So we started out very simply with just these jumbo sacks full of soil. This is a sorghum root system, and then built this frame around the outside and strung it up with fishing line, basically. We've since got a little bit more sophisticated here, and we've got a dozen of these large boxes. We've built 4 by 4 by about 7 feet. These are 8 feet, and these are 3 by 3 by about 7 feet. Filled with turfus in this case, because it's much easier to work with. Yeah, so you get, Tyler's made some slides here. So we have this internal scaffold now with PVC. You can see a drainage bed at the bottom and a tarp, so we can collect runoff and things like that. And then we outfitted these with a number of these matrix water potential sensors, and also a little tube that we can hook up to a Picaro mass spec to measure CO2, although I've learned there's better tools that we could use for that. And so the idea is we really have hourly measurements, but this is just showing you the daily measurements that we've interpolated in a model. And these are 2 different switchgrass phenotypes, an upland and a lowland, and I always forget which one's which. Maybe someone can tell me in this room, I don't know. But the idea here is to correlate the root structure with these environmental dynamics that we see over time. So we've done some really nice above ground phenotyping as well, just on tillers. I'm going to skip over this in the interest of time to get to the roots. So you can see we sort of harvest these just moving down. And you can see the heterogeneity and the complexity of the root system. And so a part I'm not going to get to show you today, but we're very interested in, for example, modeling like root cores or rhizotrons or something to really understand if the information we get, if these things are in different places, what that can tell us about the overall properties of the actual root system as it's growing. So we can see like very strong genetic differences between these 2 genotypes in this system. And here's how we turn this into data. This is using a method called photogrammetry, which is basically taking a digital camera and walking around and taking thousands of images around this box. And we can use basically algorithms, GPU enabled, that match each image. They model the camera geometry, which you're seeing here. And we get a 3-dimensional reconstruction of the entire root system. And we can apply these same tools from the X-ray or from the optical system to an entire, essentially freely grown root system. So we've been doing some water stress experiments, and you can see just the fundamental change that a -1.5 megapascal is pretty severe water stress on this genotype. The roots are much less dense, but they go really wide and really deep. I mean, we can just see it crystal clear. So this is a little bit out of order, but here's what our reconstruction room looks like. We had to put a bunch of random patterns and stuff up on the wall so that we have points that are different in each image so the camera can get modeled. And so this is a 3D reconstruction of the entire crazy room. And here where we've removed all the stuff except for the roots themselves that we can measure. So this is a process that works. It's very easy to do. So I think this is something that'll be in many of our toolboxes in the future, can be used for above and below ground. And yeah, we have a lot of good practical knowledge for that now. So I think I better stop here, thank the lab. It's a really interesting lab that we have, again, applied mathematicians, computer scientists, plant physiologists, quantitative geneticists, molecular biologists, whatever I do, I don't know, professional talkers, I guess. And so there's a couple of other quick things I want to just highlight while I have your attention. We have a new faculty search open at the assistant or associate level. So please do spread the word, or if you're in the market, please do apply. It's basically anyone with creative, innovative approaches that are related to plants. It's quite open at the center, and it's a fantastic place to work. Great colleagues, a lot of resources. We get to just do the thing that we want to do. There's really no constraints. Finally, I also want to highlight a major meeting in Missouri that's about a year from now. The 11th ISRR meeting is going to be held in Columbia. For the students and postdocs in the room, we're going to have a student ambassador program and several days at Danforth Center, career development, training, the ability to organize a pre-symposium to the meeting here. So please do put this on your calendars and take a look at it. We're still developing the schedule and everything for it, but put it on your calendars and think to come. It's a fantastic international roots and rhizosphere community. So I think I definitely better stop there. I thank you very much for your attention. Sorry for the technical stuff. That's my fault as usual. And if there's time for questions, great. If not, I'll be here and happy to chat on the side. So thank you.
Take, for example, sorghum panicle. How
Yes.
early in the development of that panicle do you strip it out, make a prediction about potential beetle?
I have the exact same question, and I don't know yet, but that is exactly the kind of question that we hope to answer with the sort of multi-scale or developmental imaging. That's exactly the kind of question that I would pose next. So I don't know, but I think we have enough information here that we should be able to do it, or maybe we can't. But I would feel confident that it's not because we didn't have a good enough representation of the actual structure in the developmental stages. If that makes sense. Yeah.
Have you tried ground penetrating radar to measure in your fields?
I have not. I'm very curious about it. I've talked with Dirk Hayes a lot, who's done the most work, and I feel like it needs to be developed more. So I think it could be useful. It's never going to give us. There's a fundamental problem with physics, like big waves penetrate deeply, but can't see tiny things. But I think what it could do, and what's probably good enough, is just give us some idea of biomass as a function of 3D space, right? And so if there's really dense regions with a lot of roots, and we coarsely know that, that's incredibly valuable, especially if we can do it over time and see these things change. So this isn't especially important because I haven't worked with the technology, but I'm holding out hope for it. Okay, well, oh, yeah.
So can you pick up interactions with organisms like mycorrhizae?
Yes, yeah, I think so. So there's kind of two ways we can do it. We can grow the thing in there and just look at how the structure changed, the macro structure. And then we can look at like very specific interactions between the two. So I think there's a lot of interesting work to do there. But again, this falls into the boat of like, I don't know very much about fungi or bacteria. And so for me, it's just finding partners who have interesting questions, interesting applications for some of the technology here. So if you've got some ideas, I'd love to hear them. Yeah.
So could like you take a picture of like the bacteria, like rhizobia, just traveling into the like nodule cells and stuff like that? Like do you like a time lapse of that?
We couldn't do we could do a pseudo time series of that. So this resolution that we get here, we need like fixed samples, and the scanning time that it takes if the movement is faster than the scanning time, it's a blur, it's a problem. Maybe in the future, when we get better with contrast enhancement, when we, there's some computational imaging methods that use like machine learning to do the imaging, maybe we'll get there, but currently, no. But what we could do is, a pseudo time series at different times post-inoculation or something, to sort of capture generally that process. For things like corn rootworm, I think we're going to be able to do that. So things that are a little bit bigger. I don't know about nematodes, but we're going to start there. We've started some corn rootworm work just to see if we can see the things sort of moving around, moving towards the roots, that sort of thing. So the thing you want to do is the hardest thing. We're going to start with something a little, something a little bigger for now. But it may be possible in the future. Maybe. Yeah. Oh, sorry.
With the larger mesocosms, has there been any attempts to look at how recent trait expression varies, like with just a single plant versus plants at yield density?
That's a great question. So, like, if we grew the same plant out a million times, like, how similar would it be, for example? Maybe not a million.
Well, I mean, like having just multiple plants within the same.
Oh, multiple plants. Yeah. Yep. So that is definitely the next vision. So the vision would be if we could take a field plot and get a box big enough. So what my current thinking is to buy a dump truck. I'm not kidding. With the detachable beds, and I think the size of that's actually was the inspiration for the mesocosm. And what I proposed to RPE, they didn't give me the $1 million for that. They gave me $50,000 to do that with. And so, okay, I think we got pretty far. So the next thing would be to do, like a much more realistic interaction. So something bridging, truly bridging the field and controlled environment situation. Thermal mass to be more similar, and water movement, obviously, the interaction. That's the next step. Yeah. Oh, sorry. Yeah.
You mentioned you had create
Yeah.
So do you have anything to 3D construction? Any free software?
We developed some, and then we use some other stuff that's more custom. So the photogrammetry, we use a software called Pix4D, which is used, it's a pretty common one. It's used for a lot of drone-based stuff, but it works really well just for freehand things. For the gel system, we developed our own 3D reconstruction algorithm that could be shared. For the Xray stuff, we mostly use the manufacturers, but we've dabbled with some other our own reconstruction methods with that. So it's like it's a mix. If it's already there, we'll use it. If it's there and it's not quite it, we'll modify it. And if it's just not there, then we have to figure out how to get it done ourselves. Yeah.
So the question, you started the answer for him with one on last
Okay.
week. So in terms of reproducibility, you talk about phenotypes, hybrids or inbreds.
Yeah.
How reproducible is that over different plants over time?
In the mesocosms or the root crowns? In the big. We haven't done enough experiments in the mesocosms yet. I mean, by eye, the two switchgrass varieties we've looked at are dramatically different. I mean, and so, they're so different that statistically they're quite distinct. Now, of course, I wouldn't base everything on one experiment, so we've got multiple rounds of these, but as you can imagine, it's a lot of work, so it's pretty low throughput. The switchgrass cells about 3 months, so we could turn these over maybe 3 times a year, maybe 4 if we were like really on top of it. I only have enough money to run a greenhouse to get 12 of them in there and a team to do it. So it's fairly slow. The same thing with the water stress. I mean, it's a dramatic difference, and so I feel pretty confident that that's not noise. In terms of the root crowns in the field, it's one of these things where if you look at any particular trait, like, okay, it's the angle or the convex holes, kind of what I was showing with the nitrogen stuff, you get a lot of variation, and some of it's probably environmental, but it's just hard to explain. You can't measure really everything out in the field. But over time, or if you look at all of the features in a multivariate space, you do get very clear genetic signals for that, right? I mean, we can discern things that look fairly similar, statistically discern them. If you look at one or two, if you look at any given trait, it's hard to do that. But if you look at a suite of traits, because they're all somewhat related, right? These aren't actually plant traits. They're a function of how we measure them or how the algorithm we write to extract the phenotype, if that makes sense. So we see a lot of environmental variation.